Article 2, the studies of abiotic components of aquatic ecosystems. Today, we are going to test the water parameter including the soft oxygen, salinity, temperatures, and also pH values of water at the stream near to, biowalk, and the buildings of faculty, fisheries, and food science. Abiotics and biotic factors work together to create a unique ecosystem, and they influence each other. To attain a quantity and qualitative aquaculture products, it's important to maintain a very good conditions of water environment. Hence, the following practical is to introduce the students with the major physical and chemical components of an ecosystem and to teach them the correct ways to measure these parameters by using the tubes. Methodologies. The materials required are the water sample from biowalks and FPSM, and a multi-parameter, and also a bucket to store water sample. First, the water sample were both collected from two different places. Then, the cover of multi-parameters removed. The apparatus was placed into both water samples and waits until the readings are stable. All the readings were recorded in table. Task 1. Plot the diagrams of physical and chemical properties. The chemical properties are from sunlight and the physical properties are from the place that plot with X. Task 2. Discuss the relations of each parameter. The water quality parameters between biowalk and FPSM are similar. The pH value and the soft oxygens are affected by the temperature. pH levels increase with the increasing in temperatures, and as the temperature increases, the soft oxygen in water decrease. Conclusion. The chemical properties such as pH, the soft oxygens, are affected by the physical properties such as temperature, and the chemical properties affected the organism in water column very much. It is obvious to see that the abiotics and biotic factors are connected to each other.
Introduction for microbenthos is organisms size larger than 1 mm. Inorganic material in sediment are called deposit feeder, such as gastropod, consists of the organism that live at bottom of a water column. Vital role in maintaining ecosystem functions such as material cycling in sediment and energy flow in food webs. Fundamental role in breaking down organic material, detoxifying pollutant dispersion, and burial. This is apparatus that we use: plastic bag, pail, tray, sediment grab, sieve, bottle, rose bengal solution for preservation. For first methodology is collect the sample using sediment grab and put the sample into plastic bags. For second methodology is put the sample into the tray after passing through 500 safe and species sorted into main group, worms, crustacean, mollusks, and others. And the last methodology is sample should be sorted in label jars and taken back to the laboratory for preservation. Samples are to be carefully labeled and preserved in a rose bengal solution. This is macrobenthos data from bio website. We got 4 species difference. And then for macrobenthos data for FPSM site, we also got 4 different species. Importance to study the benthos community in aquatic ecosystem is aquatic habitat possess a diverse range of benthic community, and they have an important role in maintaining the food web and primary production. Although most of the benthos are very small in size, they play an important role in aquatic ecosystem as they act as bioindicator, are important nutrient in different trophic level of the food chains and food web, and have a role in nutrient and mineral cycling, and they act as decomposer. Then the role of benthic macro invertebrate in cycling nutrient and controlling in nutrient outflow from ecosystem. The benthos transform organic detritus from sedimentary storage into dissolved nutrient that can be mixed into overlying water and used by rooted plants and algae to enhance primary productivity. This is Simpson index from BioWorld site. The diversity index we got 0.4272. Then the second table is Simpson index for FPSM site. We got the diversity index is 0.2601. Relationship between environmental factor with the macrobenthos is water. Macrobenthos can rapidly respond to change in water environment, which have a special advantage in pointing out the water ecological system. Then their nature of living are in sediment. They are exposed to the stressor, for example chemical pollution, Second, temperature. The temperature in an area will affect the habitat of the microbenthos if it is not compatible with its body. Macrobenthos diversity at both sampling site. For biowat site, we got 0.4272 and FPSM site 0.2601. Biowat site has the higher diversity index, so has more species richness and evenness. It would be more resistant to any environmental damage or change. And in that value 0.4 biowat site means there is a high probability that any four individuals taken from biowat site will be from different species. The areas show good species richness and evenness and more like to withstand changes such as pollutant. For the conclusion, we have done sampling into area, namely the biowat site and FPSM site. From the area, we were able to record the diversity of microbenthic communities in the area. Microbenthos are very important for the aquatic environment. We have found that the biowat site diversity index is the higher. We also found a variety of different species in the two areas.
Good day, doctor. My name is Tim Wei Ling. Weight number D61105. This I would like to present lab practical for sensitive of plankton. Phytoplankton is small cell organism source of phytoplankton. Phytoplankton use sunlight, nutrient, carbon dioxide, and water to produce oxygen and nutrient for other organisms to photosynthesis. 70% of Earth is covered by ocean. Phytoplankton is responsible to producing up to 50% of oxygen we use to breathe. Therefore, phytoplankton is a small plankton that swim freely in ocean and is the food sources for fish larvae and filter feeder organisms. So plankton is important component in marine and freshwater ecosystem. We use most massive plankton net to collect plankton from the river because the plankton is too tiny. Next, we use a compound microscope to count the plankton using Leakey method. Methodology. Apparatus have plankton net, urea potter, dropper, gas light and cover slip, and the last one, compound microscope. Procedure. We collect plankton by using plankton net from three different areas in one study site during sampling and place plankton in a urea potter. After that, we add preserved solution, which is iodine solution, into a urea potter with plankton and water. Sample analysis. First, we put one drop of sample on the glass slide and cover it with a cover slip. Next, we use compound microscope to observe plankton and count the plankton. Third, measure the cell density per ml using Leakey method. This is plankton data sheet. For pieces of plankton, it has to identify. So we just count the plankton. Result. This is a Leakey method formula that we use to measure the cell density per ml. Result. Average density of plankton at APSM is 25,572.67 cell per ml. Average density of plankton at Bearwalk is 11,812.67 cell per ml. Task 1. Why it is important to study plankton community in aquatic ecosystem? Plankton is Earth's most critical organism, so studying and understanding them is important. Phytoplankton generate about half an atmosphere oxygen source as per year as small land plant. Plankton found in all ocean ecosystems play a critical role in maintaining the health and balance of the ocean in its complex food web. In short, they make more other ocean life possible. Second, discuss the relationship between environmental factor with the plankton organism found in ecosystem. Plankton species and planktonic community vary incident by many aberrant factors such as light ability, temperature, salinity, heavy metal, pollutants, pH, and nutrient concentration, and various factors such as predator and parasite. These variations are being studied through ecological data to help policymakers. For example, where the plankton community various and harmful plankton species grow rapidly due to the excessive nutrient in the water. Third, what can you conclude about the plankton density at both sampling site? To conclude, APSM plankton density is higher than biowalk. The average density of plankton for APSM is 25,572.67 cell per ml, while the average density of plankton for biowalk is 11,812.67 cell per ml. This is because the richness of organism and APSM is more than the biowalk. That means the APSM environmental condition is better than biowalk, which allow organisms to survive and produce. Conclusion. Phytoplankton and zooplankton are important in an ecosystem because they provide food for primary consumer in the food web and maintain ecological balance.
Introductions. Group done the sampling at Jalan Bioworks on 13th February 2023 and FPSM Bridge on 20th February 2023. The aim is to sample the aquatic microsites that grow around the sample area. Methodologies: to observe and determine the aquatic microsize of submerged, emergence, and floatings at its locations. Take the picture of the aquatic microsize. Conclusions: to make a conclusion, we found that there are many different types of aquatic microsites live around our two sampling areas, which act as important habitat for other organisms to shelter them and provide food for them. To have sustainable ecosystem, we not only take care of one organism, but also need to protect aquatic microsites.
What is nekton? Nekton refers to any organisms that are capable of independent movements in the oceans. They are mostly vertebrates, and they includes fishes, cephalopod turtles, other kinds of reptiles, birds, and marine mammals. Nekton forms an integral link among primary producer, consumers, and top predators that represents a significant portion of the diets of many piscivorous birds and economically valuable fishes. Our apparatus consists of a scooping net, a bucket, filled sample containers, and buffered formalin. The procedure is very simple. We just have to collect the nekton samples by using scoop net and put the samples into separate sample bottles and label them. Lastly, we have to put the preservation solution, which is the buffered formalin. So for the task content, everything is displayed down on the nekton data sheet as portrayed on the screen. So it comprises of where the study site is, the species caught, and also the numbers of individuals. In terms of the species richness, the diversity at FPSM below the bridge is better than BioWalk, as can be seen through the higher D calculated. Whereas for the species evenness, by calculating out the relative abundance, it can be seen that FPSM have a lower deviation in terms of the value for relative abundance and hence a better species evenness. To the last part of the calculation, and that is the Simpson's diversity index, the index value indicated that FPSM even with just a slight difference of 0.01 is more diverse than BioWalk. To conclude, FPSM is better in terms of species richness, species evenness, and diversity index for their populations of nekton.
That's all. Thank you.